I think that one of the goals of diabetes research has always been how can we achieve independence from insulin. And right now, the most promising technologies are technologies that allow us to replace the beta cells that we've lost, people with type 1 diabetes or even people with type 2 diabetes. And so we need to begin to understand how we lose these beta cells in the first place. And the beta cells, of course, are the cells that make insulin. And so our research in this laboratory has always been focused on how can we ensure that these beta cells survive and not succumb to the milieu of diabetes.
So we found that before our mice develop diabetes, we see a stress on the islets, on the beta cells in particular, that is called ER stress. So it's the endoplasmic reticulum, and it is what makes your proteins, and therefore also your insulin. Right? So as the cells are making insulin, they get bogged down and they can't keep going, and then the ER becomes big and basically stops making this protein. And we call that ER stress. And we saw this in our mice now 4 to 6 weeks prior to them even developing any sort of symptoms of diabetes. And so if we can be able to target this ER stress, we can try and potentially alleviate that stress and maybe keep the immune cells from attacking it.
And I think it brings up a thought that the two diseases are maybe more closely related than we thought. And that maybe some of those drugs that have been traditionally used to treat type 2 might have some role early in type 1, even before people begin to develop the disease. And so that raises the intriguing possibility that maybe there are already drugs out there in use that may have some utility in type 1 diabetes because of this very specific kind of stress that we observe. I guess, you can get very negative about mouse studies, but the reality of it is that now, we go right from the mouse to the human. I mean, most studies are typically done in rodents first and then to humans. We don't go to intermediate animal models. They're expensive, and they're not oftentimes any more informative than the mouse. While we've cured diabetes in a mouse, what we've learned from that has actually translated to humans in many ways. A lot of the drugs that we use today have been shown to work in the mouse and therefore worked in humans. So for all the cures that we find in mice, there have been a lot of very important treatments that have resulted in humans. So I would say that anything that we do in a mouse is still exciting. Maybe what's limiting anything is how quickly we can move that into a human. So when we find something that's exciting in a mouse, then the next question is, does that give us enough information that we can do something in a human? And I think that's really what this study is about. It's suggesting that there's this process that's occurring that's been shown to happen in humans in type 2 diabetes, and we have drugs. And therefore, can now those drugs that are already proved to be applied to humans with type 1? And that's really what we're going after. So I think it's, in that respect, exciting, and hopefully will spawn some studies that we'll do here with some of our colleagues in humans. We're really not even allowed to name animals, research animals. I think the understanding is that it draws a particular connection, and they're nets, and so we don't do it. As tempting as it might be. And so I think that this is how science works, is that we don't always have a very set hypothesis when we start something. We often go in the direction that our data take us. And sometimes they take us in directions we didn't expect. And this is one of those studies where we went in the direction that our data took us, and it turned out to be something a little bit different than what we had originally conceived when we started. So, and I think that's what's fun about science is that you go with the flow, and you don't push something beyond where the data take you.